Thank you everyone. Please double tap on the screen and subscribe my channel. Those who have joined, please double tap on the screen and subscribe my channel. Thank you those who have subscribed. Radhe Sham, Radhe Sham. Those who have joined, please subscribe my channel and press the bell icon. And don't forget to double tap on the screen. Mm. It promote absorption of growth is an irreversible permanent increase in size of an organ or its part of an individual cell. In overall process, consume energy. Plant growth continues throughout the life due to the presence of meristem. Meristem cells have the capacity to divide and cell perpetuate. The growth where new cells are always added to the plant body by the meristem is called the open form of growth. The growth where new cells are always added to the plant body by meristem is called the open form of growth. Primary meristem it occurs due to the root apical meristem and shoot apical meristem. Primary growth is due to the root apical meristem and shoot apical meristem. It causes elongation of the plant along the axis. Secondary meristem in gymnosperm and dicot. It occurs due to the lateral meristem, vascular cambium, and cork cambium. It causes increase in width of the plant. So, I mean, in gymnosperm and dicot. Due to lateral meristem, vascular cambium, and cork cambium, it causes increase in the at the cellular level. Growth occurs due to the increase in the amount of protoplasm. Increase in protoplasm is difficult to measure directly, so the growth is measured by parameters like increase in fresh, dry weight, length, area, volume, and the cell number. Cell number. Maize root apical meristem can produce more than 17,500 new cells per hour. Cell size. Cell in watermelon can increase in size up to 350,000 times. Length. Growth of a pollen tube. Surface area growth in dorsoventral leaf. Oxygenate help in rooting in stem. It is widely used for plant propagation. It prevents fruit and leaf drop at early stage. It is used as herbicide. That is 2,4-D. 2,4-D used to kill dichylonous wheat but not affect the mature monocotyledonous plant. It is used to prepare wheat-free lawn by the gardener. Oxygen also controls xylem differentiation. Oxygen help in cell differentiation. Oxygen promote flowering in pineapple. They promote the abscission of the older mature leaves and mixed fruit. Apical medicine. It occurs most higher plant. The growing apical bud inhibit the growth of lateral bud. Remove of the shoot tip decapitation. It widely applicable in seed plantation and hedge making. Indole compound. Indole 3 acetic acid and indole 3 acid adenine derivative cytidine N4-furfuramine purine carnitine. In Kinetin, cytokinin, kinetin. Cytokinin have specific effect on cytokinesis. Acha, Gibberellin, so where we get more than 100 gibberellin from fungus and high plants. Widely different gibberellin helping gibberellin types are GA1, GA2, and GA3. First discovered gibberellin is GA3. GA3 acid is gibberellin acid is GA3. All GAS are acidic. Gibberellin produce a wide range of physiological response in plants. Gibberellin help or function. They increase the length of axis. Gibberellin used to increase the length of grape stalks. Gibberellin used to increase the length of grape stalks. Gibberellin elongate apple. Gibberellin elongate an apple fruit. Gibberellin improves the shape of the apple fruit. So length of oxygen is to increase the length of grape stalk. It may also delay senescence. A fruit can be left on the tree for longer period due to extended market period. Tree is used to speed up promote the melting process in beaver industries, and sugar can produce carbohydrates as the sugar in the stem. In sugar can cause spring gibberellin, the increase in length of the stem, increase the field, increase the yield as much as 20 ton per acre. When GA spraying the jubilant conifer trees, then the GA sustain promote the maturation period, thus leading to early seed production. When GA is spraying the juvenile conifer plant, then GA is sustain the mature period, thus leading to early production. Juvenile also promote bolting just prior to flowering in beet, cabbage, many rosette leaf plant. Bolting means internal elongation. Bolting occur in which plant? Beetroot, cabbage, and many rosette habitat. It means internal elongation and beet occur and many rosette plant. Cytokinesis have specific effect on cytokinins have specific effect on cytokinesis. Cytokinins were discovered from kinetin. What is kinetin? A modified form of adenine. Kinetin is modified for cytokinin was discovered from kinin. Cytokinesis has specific effect. Cytokinin have specific effect on cytokinesis. Cytokinin were discovered from kinin or a modified form of adenine. Adenine is one type of purine. Adenine is one type of purine is modified form of adenosine. Kinetin is a modified form of adenine. Adenine derivative is cytokinin. Adenine for furamine purine. What is zeatin? Isolation of zeatin from corn kernels and coconut milk because search for natural substance with cytokinesis like activities. Isolation of zeatin from corn kernels and coconut milk. Isolation of what is zeatin? Isolation from corn kernels and milk because of search of natural substances with cytokinin like activities. Several natural occurring cytokines, some synthetic compounds with cell division promoting activities have been identified. Several naturally occurring cytokinins and some synthetic compounds with cell division promoting activities have been identified after discovery of zeatin. Zeatin is a modified form of adenine, it's kind of purine. It's a kinetin, the kinetin get from the autoclaved hearing for sperm DNA. Autoclaved hearing, sperm DNA, zero from the autoclaved hearing, and then kinetin we get from autoclaved. Then function, cytokinin function, in region areas where natural cytokinin are synthesized, cell division occur rapidly. Cytokinin function or in regions where natural cytokinin are synthesized, cell division occur rapidly. Shoot bud young fruit. It helps to produce new leaf. Helps to produce chloroplast in leaf. It helps to produce lateral shoot growth. It helps to produce adventitious shoot formation. Help cytokinin help overcome the apical meristem. Cytokinin help to overcome the apical meristem. Cytokinin promote nutrient mobilization. Cytokinin promote nutrient mobilization. Cytokinin helps in develops leaf senescence. Gibberellin also help in delay leaf senescence. Cytokinin help in delay leaf senescence. Cytokinin promote nutrient mobilization, overcome apical meristem. Ethylene is a simple gaseous PGR. Ethylene synthesized, ethylene bad, ethylene synthesized. It's helping ripening food, large amount of tissue under senescence, undergoing senescence. It helps in ripening food and it helps in ripening food, large amount of tissue undergoing senescence. Influence of the Italian plant, horizontal growth of the seedling, swelling of the axis, apical hook formation of the dicot seedling. Influence of the Italian the horizontal growth of seedling. Horizontal growth of seedling. There have been horizontal growth of seedlings, swelling of the axis. Horizontal growth of the seedlings, swelling of the axis. It had an influence of Italian horizontal growth of seedling. Horizontal growth of seedling, swelling of axis, apical hook formation in dicot seedling. It promotes senescence and abscission on plant organs, especially of leaves and flower. Ethylene promotes senescence. Gibberellin delays senescence. Cytokinin delays senescence. Sene- Senescence promote and delay gibberellin GF3 cytokinin promote ethylene uh, abscission on plant organs, especially of leaves and flower, especially of leaves and flower. It promote abscission of older mature leaves and fruit. It promote maturation means auxin promote abscission of older mature leaves and fruit. Promote abscission of older mature leaves and abscission of older mature leaves and fruit. Promote the abscission of older mature leaves and fruit. And ethylene promote abscission of leaves and flower. It promote rapid internal elongation, petiole elongation in deep water rice plant. It promote root growth and root hair formation. Root growth and root hair formation. It promote female flowering in cucumber by thereby increase. So ethylene is a simple gaseous PGR. Ethylene helps in ripening of food, large amount of tissues undergoing senescence. Ethylene is on plant, horizontal growth of seedlings, swelling of axis. Horizontal growth of seedlings, swelling of axis, apical hook formation in the dicot seedling, apical hook formation. Horizontal growth of seedling, swelling, and apical root formation in dicot of seedling. It promotes siliceous and abscissions of plant organs, special leaves and flower. And in auxins, it promotes abscission of mature plants and fruit, mature plant leaves and fruit. Yes, It promote root growth and root hair formation, but in auxins it promote root differentiation. That's written. Initiate rooting in stem cutting. Initiate rooting. Sorry. Initiate rooting in stem cutting. Initiate rooting in stem cutting. Okay, ethylene help is highly effective in fruit ripening. Increase the respiration rate during fruit ripening. Increase the respiration rate. The increase in respiration rate is called respiratory climate. Ethylene breaks seed and bud dormancy, initiate germination in peanut seed, help in sprouting of potatoes tuber. Ethylene help leaves or upper part of the shoot to remain above water. Ethylene help to increase their absorption surface in their plant. Ethylene is used to initiate flowering and synchronizing fruit in pineapple. Auxin also promote flowering. Ethylene also initiate flowering and for synchronizing fruit set in pineapple. Ethylene also induce flowering in mango. Flowering in mango. Ethylene regulates so many physiological processes, hence is one of the most widely used feature in agriculture. So ethylene is gaseous feature. It's help in ripening of food, large amount of tissue undergoing senescence, horizontal growth of seedlings, swelling of the axis, apical hook formation, dicotyl seedling. It promotes senescence and abscissions on plant growth, especially leaves and flower. It promotes rapid elongation petioles in deep water sea plant. It promotes root growth and root formation. It promotes female flowers in cucumber, initiate flowering in pineapple, respiratory it increase the rate of respiration, that is respiratory climax. Ethylene breaks seed and bud dormancy. Ethylene also induces flowering in mango. Ethylene also induces flowering in mango. Ethylene regulates so many physiological factors, hence it is the one of the most usually used feature in agriculture. Uh, 84 is widely used compound for ethylene for the source. It is widely used compound for the source of ethylene. 84 is an aqueous solution. Is readily 84 is in an aqueous solution is readily absorbed and transported within the plant and release ethylene slowly in an aqueous solution is readily absorbed. 84 Huston fruit ripening in tomatoes and apple. Sure. 84 Huston fruit ripening in tomatoes and apple. Ethylene helps in initiating in flowering in pineapple and helps in sprouting of potato tuber. Help in potato tuber. Tree T and H making by auxin. Okay. Promote flowering in cucumber also ethylene. Sure. Ethylene accelerate abscission in plant flowers and fruit, cherry and walnut. Example, cherry and walnut. Fruits and flowers and fruit. Water is an essential for cell enlargement. Trajectory of cells help in extensions of growth. Water provide medium for enzyme activities needed for growth. It helps in release metabolic energy for growth. Macro and micro elements are needed for the synthesis of protoplasm and hence act as a source of energy. Loss of protoplasm to form tertiary elements, they also develop as dominant to carry water. Loss of protoplasm. The loss of protoplasm to form matter. I'm reading biology, so those who have joined, please subscribe my channel and press the bell icon. Meristematic cell plasmatic growth differentiation and matures sequenciation, mature cell expansion, maturation. Those who have joined, please subscribe my channel and press the bell icon. Heterophyly due to phase of life cycle in cotton, coriander, and lemon. Heterophyly due to phase of life. Example: cotton, coriander, larkspur. The leaves of the juvenile plant and mature plant are differentiated in shape. Heterophyly due to environment is buttercup. Heterophyly due to environment, buttercup. Heterophyly due to heterophyly in the phase of life. Heterophyly in the phase of life, cotton and coriander. Heterophyly due to phase of life, cotton and coriander. Heterophyly due to phase of life, cotton and coriander. Heterophyly related to environment. Buttercup, heavily due to environment buttercup. Heavily due to the phase of life, cotton and coriander. Acha. Factor controlling the development. Intrinsic factors include intercellular genetic, intracellular genetic. Intercellular factors such as growth regulator. Intracellular genetic, intercellular. Extrinsic factor like temperature, water, meaning guys, let me drink water. I'm thirsty. Abscisic acid, abscisic acid, abscissions and dormancy. It also has other wide-ranging effect on plant growth and development. It acts as a general plant growth inhibitor. AB inhibit act as a general plant growth inhibitor. It inhibited plant metabolism. AB inhibits seed dormancy. ABS stimulate closure stomata in epidermis, increase the tolerance of the plant to various kinds of stress. ABA play important role and control. ABA control regular seed development. ABA regulate maturation and dormancy. ABA control regulates seed development. ABA control or regulates seed development. ABA control and regulate seed development. ABA regulate maturation dormancy. Function of ABA. It by including dormancy, ABA helps it to withstand desiccations and other factors of growth. By inhibiting dormancy, ABA helps it to withstand desiccations and other factors of growth. ABA acts as a antagonist to GAS, that is gibberellic acid. Okay, this chapter complete. Those who have joined, please subscribe my channel and press the bell icon. I've completed so differentiation, two typical suitable differentiation intersection. For senescence promoted by ethylene is delayed by GAS and cytokinin. Glycolysis or EMP pathway. Glycolysis, that is the EMP pathway. Glucose is a Greek word. Glycolysis. Glycose means sugar, lysis means splitting, and this term is known as glycolysis, given by Emden, Merov, and Farnes. Glycolysis anaerobic organisms, anaerobic glycolysis by anaerobic organisms, no oxygen. Glycolysis occur in the cytoplasm or cytosol of the cell. It present all living organisms. Here, glucose undergo partial oxidations, no CO2 produced. Glycolysis undergo partial oxidations to produce 2 into pyruvic acid, that is 3 carbon. Pyruvic acid is 3 carbon. In the pathway, glycolysis occur in an organism. Glycolysis occurs in cytoplasm or cytosol of the plant. It present in all living organisms. Cell glucose undergo partial oxidations. Glucose undergo partial oxidation to form 2 into pyruvic acid, that is 3 carbon. Glucose derived from sucrose. In product of photosynthesis, glucose in the which are transported to root cell. Sucrose by invertase enzyme. Glucose plus fructose. Glucose is monosaccharide. Fructose is also a monosaccharide. Glucose by invertase enzyme form sucrose by invertase enzyme forms glucose and fructose. Glucose, sucrose, invertase enzyme, glucose plus fructose. Glucose, plant lipids. Sucrose. Total step in glycolysis is 10. ATP utilizing step, that is 1 and 3. Step 1 and 3. 2 ATP. Step 1 and 3. And ATP formation step 17. From ATP utilization 1 3. Formation step. ATP formation step 7 and 10. Here 2 ATP utilized and here produced 4 ATP. Net gain ATP is 2 ATP. Reversible Irreversible is 137. Irreversible step is 137. Net or final net product of glycolysis is ATP2 and ATPH2 pyruvic acid 2. Net or final product of glycolysis ATP and ATP pyruvic acid. ATP2 and ATPH2 pyruvic acid 2. ATP2 and ATH2 pyruvic acid 2. ATP2 and ATH2 pyruvic acid 2. CO and O2. Glucose 6 phosphate. Glucose 6 phosphate. Fructose 6 phosphate responses form of 1,6 biphosphate. Glucose 6 phosphate. Glucose 1,6 biphosphate. Then trisphosphate. Dihydroxyacetone phosphate. Step 5 glycerylate 3 phosphate. That is trisphosphate. 1, comma 3 biphosphoglyceric acid, 2 into triphosphate, 2 into tris biphosphate, NAD plus, NADH plus, NAD plus, NADH plus, then 2 into triphosphate, 3 phosphoglyceric acid, then 2 phosphoglycerate, 2 phosphoenol pyruvate, pyruvic acid, glucose, glucose 6 phosphate, glucose 1 6 biphosphate, triphosphate, dihydroxyacetylene phosphate, glycerate 3 phosphate, then 1 comma 2 biphosphoglyceric acid, 1 comma 3 biphosphoglyceric acid, 2 into tris by 1 comma 3 biphosphoglyceric acid, group of NAD plus, take NADH plus, triphosphate 3 phosphoglyceric acid, 3 phosphoglyceric acid, 3 phosphoglyceric acid, 1 comma 3 biphosphoglyceric acid, 3 phosphoglyceric acid, step 7. ATP, and step 7 is the step of ATP formation. Step 8, 3 phosphoglyceric acid, phosphoglycerate, pyruvate, phosphoenol pyruvate, and pyruvic acid. Pyruvic acid is the key product of glycolysis. Its metabolic rate depends on the cellular need. Its metabolic rate depends on the cellular rate. Glycolysis indirectly ATP, oh no, indirectly ATP, directly 4 ATP. Nearly is to SLP. SLP 2 ATP formation and oxidative phosphorylation 6 ATP. SLP 2 ATP formation, oxidative phosphorylation 6 ATP. So kinase ATP fermentation, pyruvic acid 3 ways. Lactic acid fermentations, alcoholic fermentations, and aerobic fermentations. Lactic acid fermentation and alcoholic fermentations are anaerobic respiration. Glycolysis is anaerobic with partial oxidation. Fermentation, alcoholic and lactic acid, they are anaerobic respirations and incomplete oxidation and occur in prokaryotes and unicellular eukaryotes. Complete cytoplasm and cytosol. Aerobic respiration, complete oxidations. Two type of fermentations and aerobic respiration. Alcoholic fermentations and lactic acid fermentation. গ্লুকোজ থেকে পারুভিক অ্যাসিড থেকে NADH প্লাস থেকে reducing agent NADH plus NAD plus ethanol plus CO2. Here enzyme alcohol fermentation, pyruvic acid decarboxylase and alcohol dehydrogenase. Pyruvic acid decarboxylase, alcohol dehydrogenase. Alcoholic fermentation example is yeast. Yeast poison themselves to death when the concentration of alcohol reaches about 13%. Alcohol fermentation, glucose থেকে pyruvic acid, ethanol plus CO2, ethanol. Enzyme used pyruvic acid decarboxylation, pyruvic acid decarboxylations and alcohol dehydrogenase. Alcoholic fermentation example is yeast. Yeast poison themselves to death when the concentration of alcohol reaches over 13%. Lactic acid fermentation, lactic acid reduce NADH plus to NAD plus. এখানে রিডাকশন হচ্ছে, এখানে রিডাকশন হয়ে এখানে এনজাইমটা ইউজ হচ্ছে ল্যাকটেট ডিহাইড্রোজেনেস, alcoholic fermentation, alcoholic dehydrogenase, and pyruvic acid decarboxylase. Pyruvic acid decarboxylase and alcoholic dehydrogenase. Lactic acid dehydrogenase. The reducing agent is ADDH plus H plus is reoxidized to NADH plus in both process. In animals, when the oxygen is inadequate during exercise, pyruvate acid in the muscle is reduced to lactic acid by lactate dehydroxy enzyme. When oxygen is inadequate during exercise, pyruvic acid in muscle cell is reduced lactic acid. By lactic dehydrogenase enzyme. Less than 7% energy is released. Less than 7% energy is released from glucose during lactic acid fermentations and alcoholic fermentation. Less than 77% energy is released from glucose during lactic acid fermentations and alcoholic fermentation. Lactic acid fermentation example is some bacteria, and alcoholic fermentation example is yeast, and is poison themselves to death when the concentration is over 30% and energy produced less than 7%. Here In lactic acid fermentation, the enzyme used is lactic decarboxylase and lactic dehydrogenase. Lactate dehydrogenase and alcoholic dehydrogenase and pyruvic acid decarboxylase. So, net ATP production in fermentation is 2 ATP, 4 ATP from glycolysis. Both the fermentations are not release much energy. Release less than 7% energy glucose is released and not all of it trapped as a high energy of it. Hazardous process is acidic or alcohol are formed. This product, acid or alcohols are formed. Now, aerobic respiration. Those are present, please. Subscribe this channel and press the bell icon. Those are present, please subscribe this channel and press the bell icon. Those who have joined, please subscribe my channel and press the bell icon. Aerobic respiration is a complete oxidation process. Organic aerobic CO2 H2O energy. Aerobic respiration occur in mitochondria, but glycolysis occur in cytoplasm or cytosol. Matrix link reaction between glycolysis and Krebs cycle. Glucose form pyruvic acid. Oxidation NAD plus reduction. Those who have joined, please subscribe my channel and press the bell icon. Oxidation NAD plus to NADH plus or acetyl carboxylase. Krebs cycle. Product is NADH, CO2. Okay, 2 CO2 to NADH. Two important steps in aerobic respiration: complete oxidation of pyruvic as pyruvate and passing on electrons, that is ETS cycle. Stepwise removal of all hydrogen atoms, leaving three CO2 molecules. It takes place matrix of mitochondria. So complete oxidation of pyruvic acid takes place in the matrix of mitochondria and stepwise removal of all hydrogen atom. Glucose 2 pyruvic acid oxidation NAD plus থেকে NADH plus. It occurs in our membranes of the mitochondria, remove as a part of hydrogen atom, remove as a part of hydrogen atom to molecular O2 with simultaneous synthesis of ATP. Remove as a part of hydrogen atom. Remove as a part of hydrogen atom to molecular O2. Remove as a part of hydrogen atom to molecular O2 with simultaneous synthesis of ATP. Remove as a part of hydrogen atom. Remove as a part of hydrogen atom with molecular O2 with simultaneous synthesis of ATP. Oxidative decarboxylation, that is the link reaction. The link reactions occur between the glycolysis and Krebs cycle. To pyruvic acid COA plus NAD plus pyruvic decarboxylase Mg2 plus to pyruvic acid COA NADH to acetyl coenzyme CO2 NADH to NADH CO2 to acetyl coenzyme. Pyruvic acid CONAD plus. Product of oxidative decarboxyl acetyl coenzyme CO2 NADH plus acetyl coenzyme CO2 NADH acetyl coenzyme CO2 NADH plus coenzyme CO enzyme. So link reaction to NADH to NADH2, that is 6 ATP. 1 NADH means 3 ATP. Total Krebs cycle. Krebs cycle. Those who have joined, please subscribe my channel and press the bell icon and double tap on the screen. Krebs cycle, that is TTC cycle, oxaloacetic acid, acetyl coenzyme plus water. Oxaloacetic acid, acetyl coenzyme plus water. CIO alpha SSFM. CIO, C for citric acid, I for isocitrate. CIO alpha SSFM. CIO alpha SSFM. Citric citrate synthase. Citrate synthase. Citric acid to isocyanate. Isocitrate. Citric acid isocitrate. Citrate synthesis. Citrosynthesis. Citrate synthesis. Citric acid. CI. Isomerase O for oxidation, that is oxidation. O for oxidation. MB is formed. O alpha. Alpha decarboxylation, CO2 removed. Decarboxylation, CO2 removed. Decarboxylation, CO2 removed. NAD is formed. Mm. GDP. Here, no oxidation occur. One is the succinic acid, FADH2, FADH2, fumarate, malate, oxaloacetic acid. Fumarate, malate, oxaloacetic acid. Fumarate, malate, oxaloacetic acid. Fumarate to malate, water released. अच्छा oxidation FADH form. CIO, citric acid, isocyanate, oxalosuccinate. Here oxidation is formed. Alpha ketone, glutaric acid, decarboxylation, CO2 formed, CO2 removed. Fumarate to malate. NADH2, NADH, FN, complex 3 to cytochrome B, FES, cytochrome 1. Complex 2, FAD, FES. Complex 4, copper A, cytochrome A, copper A3, cytochrome B. Copper A, cytochrome A. Copper A3, cytochrome B. Complex 3 there, cytochrome BC 1. complex 1 NADH dehydrogenase, complex 3 complex B1, complex 2 succinate enzyme name succinate dehydrogenase, complex 4 cytochrome C oxidase, copper A copper A cytochrome A, copper A cytochrome A, copper A cytochrome A3 copper B, copper A cytochrome A, then cytochrome A3, and complex 4 ATP formation state. Complex 1 NADH formation. NADH dehydrogenase. Complex 1 enzyme NADH dehydrogenase. Complex 3 cytochrome BC1. Complex 2 succinate dehydrogenase. Complex 4 cytochrome C oxidase. A4 is moved down through which proton? Carbohydrate 1.0, fatty acid less than 1. Carbohydrate 1.0, fatty acid less than 1. Fatty acid break down acetyl coenzyme before entering into respiratory pathway. During breakdown, catabolism. Putting the amino acid glycogen. So up to this guys, bye bye everyone.